Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjon classes. Dear student, we are studying a plant physiology and we are in a continuation of the chapter that is photosynthesis in higher plant. In the previous class, in the previous two classes, we have already talked about the, we can say, the part of early experiment and introductory part, what is photosynthesis, that we have discussed. And in the previous class exactly we have discussed about the different we have talked about the site of photosynthesis, that is chloroplast, how the pigments are there, where what which pigments are there, how they are located, all these things we have discussed. Now in today's class we are going to start with the photosynthesis. Okay, we are going to start with the photosynthesis, and in that we are going to talk about the mechanism of photosynthesis, and we will be going to talk about the light reaction. So let's talk about light reaction in detail. Okay, so this is the first phase of photosynthesis. We have understood that it is divided into two phases. Whole photosynthesis is divided into two phases. First is light reaction and another in the dark reaction. In this lecture, we are going to talk about the light reaction mainly. Okay. So first and foremost question is that what is light reaction? Okay. So simply, if we understanding this term, light reaction means anyway light is involved, and we know that light is essential for the photosynthesis. So light reaction, which is also known as photochemical phase, which include light absorption. So in light reaction, what is going to take place? First, light absorption is going to take place. Apart from that, you will see that water splitting is taking place. And if water is splitting occurring, then oxygen release you will see. And you will see that there is ultimately formation of high energy chemical intermediates, that is the ATP and NADPH in this light reaction, that you will see. Okay, in this particular thing. So now, ultimately, the goal of this light reaction is to synthesize ATP and NADPH2 as a reducing power that is going to use in the dark reaction, which is a reduction phase. And this light reaction is a one type of oxidation phase. Okay, so this many events are going to take place in this light reaction. Let's discuss how they take place. First, for the light absorption, there is a need of pigments. Yes or no? There is a need of pigments because pigments are going to carry out that function. So the pigments are organized into two discrete photochemical light harvesting complexes. They are here arranged in the two discrete units, and these two discrete units are the nothing but light harvesting complexes. Means light harvesting complexes include the group of pigments within the photosystem 1 and photosystem 2. So each photosystem, that is the photosystem 1 and 2, we have discussed in the previous class. They are described based on their origin, and these both photosystems have their light harvesting complexes. Now light harvesting complexes along with the reaction center going to form a photosystem. Now the light harvesting complex is not only made up of pigments, it is made up of hundreds of pigments molecules bound to the proteins. So proteins are also there in the light harvesting complex. Now here for the light harvesting complex, the term antenna is used. An antenna in each photosystem has all the pigments forming a light harvesting system called an antenna. Each photosystem has all the light harvesting system pigments which are called antenna. Means if we exclude chlorophyll A or reaction center from the photosystem, you will get this antenna or a light harvesting complex. Now, what is the function of this light harvesting complex or the antenna molecule or the accessory pigments? Their function is to particularly, ultimately accept the discrete wavelength of light. Okay, these are the pigment molecules, which is basically accessory pigment molecules, and this light harvesting complex. These pigments absorb a photon and ultimately passing is to the reaction center, and from reaction center the magnesium of chlorophyll A, which is present in reaction center, get excited and it loses electron from its outer orbital. From where? Outer orbital of the magnesium, which is present in the chlorophyll A of reaction center. So ultimately these light harvesting pigment are playing a role in what? Taking a various wavelength and ultimately giving it to the reaction center, that is chlorophyll A. Okay, and this whole is a light harvesting complex. Okay, here we can say this is the, we can call it as a reaction center, and this is a primary acceptor of this reaction center or photosystem. Now these pigments, specifically accessory pigments, help to make a photosynthesis more efficient by absorbing different wavelength of light. So these pigments help to make a photosynthesis more efficient by absorbing different wavelength of light. Means this accessory pigment or light harvesting pigment have a role to absorb different wavelength of light or discrete wavelengths of light. And a special chlorophyll A form the reaction center. The reaction center is different in both the photosystem, as we have discussed already in the previous class. In PS1, the reaction center is chlorophyll A that has a absorption peak at 700 nanometer. Means its the absorption peak maxima is 700 nanometer. Hence, it is called a P700. And in PS2, it has a absorption maximum at 680 nanometer. So we call it as a P618. Okay. So this both photosystem have a two different reaction center. Yes, they have chlorophyll A, but the absorption maxima is different. Now, so in this particular thing, we have to discuss light reaction. We have to mainly discuss the non-cyclic and cyclic photophosphorylation, because in light reaction, non-cyclic and cyclic photophosphorylation take place, and through that only ATP and NADPH2 synthesis occur. So in non-cyclic photophosphorylation, first we have to understand what is phosphorylation. So as we know, the process, okay, through which ATP is synthesized from the ADP and inorganic phosphate, we call it as a what? A phosphorylation. So it is just formation of ATP by this from the ADP and inorganic phosphate. Now another question is, phosphorylation is formation of ATP from the ADP and inorganic phosphate. ATP is adenosine triphosphate, ADP is adenosine diphosphate, and PI is inorganic phosphate. So when diphosphate react with another phosphate, it is going to form a ATP. But when a term photo comes in the suffix, in the prefix of this phosphorylation, then it is called as photophosphorylation. Now the question is, what is photophosphorylation? Then photophosphorylation, if we talk, it is a process through which ATP is synthesized from ADP and inorganic phosphate. Obviously, why? Because the phosphorylation term is used, but photo term is used because this phosphorylation of ADP and formation of ATP is occurring in the presence of light. Now, in non-cyclic photophosphorylation, we will see that the two photosystem work in a series, first PS2 and then PS1, and the process completely called as a non-cyclic photophosphorylation. Why it is called as non-cyclic photophosphorylation, we will understand in detail. And the two photosystem are connected through an electron transport chain. These two PS1 and PS2 are connected through electron transport chain as seen in the Z scheme. Z scheme means non-cyclic photophosphorylation. Why non-cyclic photophosphorylation is known as Z scheme, that also we will understand in the next slide. So let's discuss how non-cyclic photophosphorylation take place. Now it is known that in non-cyclic photophosphorylation, two photosystems are involved, PS2 and PS1. Suppose I am drawing here PS2 and here I am drawing PS1. This PS2 has its accessory or light harvesting pigment, which are absorbing different wavelength, and ultimately they have a reaction center that is P680, because it is a P680 is nothing but chlorophyll A 680. It has a absorption maximum of 680. Here also there are accessory pigments, and they have P700, that is, or we can say chlorophyll A700, because they have an absorption maximum of 700. So this P680 and P700 are reaction center. Now when they absorb a light, these both pigment are going to absorb a light. This wavelength which are absorbed, they are passes ultimately to reaction center. Now, as I told you, reactions under that is P680 and P700 both have a magnesium in their chlorophyll. Now, when light energy is reaching to the magnesium, it carry out a electronic excitation, and two electrons are removed from the outer orbital of the magnesium of this chlorophyll A or reaction center of PS2. Same way, two electrons are lost or removed from the outer orbital of the, we can say, outer orbital of the chlorophyll magnesium of chlorophyll A of PS1. When this both electron losses from the outer orbital of magnesium of both this reaction of PS2 and PS1 respectively, it is known as electronic excitation of this electrons. Now these electrons are moving from this magnesium of this reaction center, then the two electrons are deficient here in the magnesium of this chlorophyll A. And same here also in P700 also magnesium has two deficient electrons. Now this electron which is moved due to the electronic excitation from the reaction center of PS2 that is received by the pheophytene. So here pheophytene act as a primary acceptor, primary electron acceptor of PS2. Same way, these two electrons which are released from the P700 or magnesium of chlorophyll of PS1 that is received by the iron sulfur protein. This iron sulfur protein is also a primary electron acceptor of PS1. Further, the electron which is accepted by this primary electron acceptor of PS2, that is pheophytene, that is pass on to plastoquinone. From plastoquinone, they are ultimately passes to cytochrome B. From cytochrome B, it is passes to cytochrome F. From that, it is passes to electron to plastocyanin. And from plastocyanin, the two electrons are ultimately reaching to the magnesium of a chlorophyll of PS1. Means the deficiency which was occurred due to the loss or electronic excitation was fulfilled by the electron which is received from this plastocyanin electron carrier. Okay, now be remember here PQ is plastoquinone, CYT is cytochrome, and here two cytochromes are there, cytochrome B and cytochrome F, cytochrome B6 and cytochrome F actually, and PC is the plastocyanin. Now the deficiency of the two electron in magnesium of a reaction center of PS1 is completed by the electron received from the PS2 or a reaction center magnesium of a reaction center of PS2. Now what is about the electron which is lost from the reaction center of PS1 due to the electronic excitation? These electrons are received by the iron sulfur protein. Further they are passed on to ferredoxin, that is FD. So what is FD? FD is ferredoxin. From ferredoxin, these two electrons are passes to the NADP. Now NADP plus take this two electron and 2H plus from the stroma and synthesizes NADPH2. So here NADP get reduced by the accepting two electron from the ferredoxin and reacting it with 2H plus, it is get reduced into the NADPH2. So here the enzyme which is used, that is NADP reductase. NADP reductase enzyme will work. Now here the electron which is lost from the reaction center of PS1 that is used in the NADP reduction, the electron which is lost from the PS2 that are ultimately completing the deficiency of electron in the magnesium of reaction center of PS1 or chlorophyll of PS1. And during this, be remember, during this electron transport, this pheophytene, plastoquinone, cytochrome B6, cytochrome F, plastocyanin, all are electron carriers. So during this transport, it is known that near about plastoquinone, some energy is getting released, and that is used in the condensation of ADP and inorganic phosphate, and that lead to the formation of ATP. So here during non-cyclic photophosphorylation, ATP is also synthesized during the electron flow. Okay. So now from this we are clear that several electron carriers are involved, and in that the electron which is lost from the reaction center of PS1, that is used in NADP reduction or NADPS2 synthesis, and the electron which is lost from the PS2 is fulfilling the deficiency of reaction center of PS1 along with ATP synthesis is taking place. And now what will happen to the deficiency of magnesium of a reaction center of this PS2 or chlorophyll of PS2? Now that is also completed, but that is completed by a breakdown of water, and this water breakdown will occur, that is release 2H plus 2 electron and half photo. So this half photo will be released outside, 2 electron are accepted by this one, so deficiency of this get completed and 2H plus are released. Now during this so during this breakdown of water and passing of electron to the reaction center of PS2 and release of half O2, three we know that three mineral elements are involved in this, that is manganese, chlorine, Mn plus 2, chlorine and calcium. These three elements are involved. And here during this breakdown of water, light is involved. So if light is involved here, this whole mechanism of evolution of O2 and breakdown of water is known as photolysis of water. And here the enzymes and carriers which are involved here, that all will be named as OEC, that is oxygen evolving complex. So this consists of what? Enzymes and proteins which are involved during this breakdown of water or photolysis of water. And due to this photolysis of water, the deficiency of this reaction center get completed. And here you can see that electron when moving from PS2, it is not coming back to the PS2. Electron moving from water is not coming back to the water. Electron moving from PS1 is not coming back to the PS2. Because of this, the flow of electron is non-cyclic. The flow of electron is non-cyclic. Because of this, it is known as non-cyclic photophosphorylation. And ATPase is also getting synthesized and light is also involved. Because of this, we call it photophosphorylation. Now, dear student, here you can see it is somewhat jet-like. Because of this, this non-cyclic photophosphorylation is also known as Z scheme. It is also known as Z scheme. Okay, I hope you understood it. Just I'm giving you a few seconds, look over it, understand it, then we will move further. Or you can understand it in more detail by pausing it. Okay, you pause it and understand it. So after understanding this non-cyclic photophosphorylation, we have to get through a further thing. So here in non-cyclic, by non-cyclic photophosphorylation understanding, what we understood. So we understood that in non-cyclic photophosphorylation, we can say that both PS1 and PS2 are involved. What we can note down further? We can note down that apart from involvement of both PS1 and PS2, both ATP and NADPH2 are synthesized. Apart from this, you will see that here photolysis of water take place, and here photolysis of water is taking place. Because of this, we can say oxygen evolution occur. So these things we can note down by understanding this non-cyclic photophosphorylation. Now, after understanding this and non-cyclic flow of electron, both PS1, PS2 are involved, ATP and ATPS2 are synthesized, photolysation of water is taking place, oxygen evolution is occurring. Now we have to note down about the location. And if you want to talk about location, both PS1 and PS2 are involved. So at a time, both PS1 and PS2 can be present on the granular thylakoid. On stromal thylakoid membrane, you will not see both PS1 and PS2 because on the stromal thylakoid membrane, you will see only PS1. But both PS1 and PS2 at a time present on the granular thylakoid membrane. So location is granular thylakoid membrane. Here, this thing take place. Okay, so the location of this non-cyclic photophosphorylation is the membrane of the grana thylakoid. Why it is? Because both photosystems are located there, and we call it as also a Z scheme. Okay, so dear student, we remember one more important point that Z scheme or non-cyclic photophosphorylation was proposed by Hill and Bendar. So Hill and Bendar proposed this. Okay, so this is the thing. Now, in this non-cyclic photophosphorylation, we have discussed that there is a particularly a photolyzer water. Yes or no? We have discussed. So now the question is how this photolyzer water actually occurring that we have to discuss. So if we talk about the photolysis of water, where the light is involved in the breakdown of water, this photolysis of water occur at grana, occur at grana, but on lumen side of grana thylakoid membrane. Suppose this is the thylakoid and this is thylakoid membrane. So what is the space enclosed within it? That is a lumen. And this is thylakoid membrane. So this photolysis occur on the lumen side of grana thylakoid. Okay, grana thylakoid. Because so what non-cyclic photosynthesis occur on the this side or in the this particular grana thylakoid only. And as we have discussed, three minerals are involved. Okay, that is manganese, calcium, and chlorine are associated with photolysis of water. Now, when photolysis of water occur or splitting of water occur, actually initially four water molecules get involved. We are writing there only water, but here four water molecules actually initially get involved. And with the involvement of oxygen evolving complex, that is the enzyme, which is the protein, that get broken into the 4H plus and 4OH minus. Now this 4H plus and 4OH minus, in that this 4OH minus that further with the help of OEC get broken up into 4 electron plus O2 plus 2H2O. So oxygen is released from this 4OH minus. Now, so from two more water molecules are ultimately regained. So if we summarize this above reaction, then we come to know that summarized equation if we write, then it is 2H2O with the help of which OEC, it is breaking up into the 4H plus 4 electron and O2. So if we write it for only one water molecule, then it is going to be 2H plus 2 electron plus half O2. Because of this, we are writing ultimately this ratio. Okay. But actually, if you talk 4 water molecule involved and 2 water molecule get regained. So ultimate reaction we write, because of this we write half O2, but for one oxygen evolution, actually 2 water are involved. Okay. So this way the whole process takes place in the photolysis of water. Okay. Now here for photolysis of water, the oxygen is evolved obviously, but light is required. So how much light is required? That is the question. So there is a one more thing we are going to understand here, that is a quantum requirement. One more term is quantum requirement. And if we talk about a quantum requirement, it is the number of light quanta. It is the number of light quanta or photon required for the evolution of for the evolution of one molecule of oxygen in photosynthesis. So quantum requirement is the number of light quanta or photons required for the evolution of one molecule of oxygen in photosynthesis. So Emerson was the scientist who calculated that. Emerson calculated the quantum requirement is 8. Means 8 quanta of light is required for the evolution of one O2 molecule. So quantum requirement for the evolution of one oxygen molecule is the 8. But now after 8 quanta is involved in evolution of one oxygen, then what is the per quantum yield? So for that, if we calculate the next thing we discuss here, that is quantum yield. And quantum yield tells that the number of oxygen molecule, it describes the number of oxygen molecule evolved by one quantum of light, evolved by one quantum of light in photosynthesis. In photosynthesis. That is called quantum yield. So the number of oxygen molecule evolved by one quantum of light in photosynthesis is called as quantum yield. So hence, if we talk about quantum yield, then it is for one evolution of one oxygen molecule, 8 quanta of light are required. So this is going to be for one oxygen molecule. So a number of oxygen molecule per quantum. So 1 by 8 if we talk, that it is going to be 0.125 or 12.5% of oxygen molecule is evolved per quantum of light. So that is the quantum yield. Okay. So this is what about the particularly we can say the photolysis of water and non-cyclic photophosphorylation. Now in that also one more concept was explained by the immersion. Emerson told about this quantum requirement in quantum field, but he also done an experiment. So if we want to understand this experiment, then we call it as a immersion effect. We call it as a immersion effect or and ray drop. Then if we talk about immersion effect and ray drop, dear student, it is known that immersion and Arnold was a scientist who experimented upon chlorella, and he showed two things. One is a ray drop and one is the immersion enhancement effect. What was their observation while studying the photosynthesis in chlorella? He showed that when we give a particular wavelength of light and we talk about the photosynthesis, photosynthetic yield, he showed that when we give a greater than 680 nanometer or 700 nanometer wavelength, the photosynthetic rate was observed that was this much. But when we given both 680 nanometer and 700 nanometer wavelength, the photosynthetic yield was this much. Means the yield can be this much, okay, but it was not this much, and this much less. This much less is that is due to the only the light which is given above 680 or 700 nanometer. But when we supplemented this 700 nanometer with the 680 nanometer, this much increase was observed. Okay, so in initial first experiment, this is a downfall. This is not a, it is not reaching this much. Means this is a drop actually, but it has a capacity that it can increase, but it can increase when we supplement with the 700 nanometer, the 680 nanometer. So if we apply a greater than 680 nanometer resonance, means we are applying a wavelength which is required for the PS1. And here if we are applying, then this is for PS2 and this is for PS1. So this what explain. Then we are giving a red light continuously after a certain period of time. It is oxidizing the pigment. Because of this the drop is occurring in the photosynthetic. We can say the yield. But when we applying another one, that is a 700 680 nanometer, also the yield is increased. And these two things was explained as a red drop and enhancement effect. So this is explained as a red drop, and this was explained as a immersion enhancement effect. So if we write it, what we can write? Red drop and immersion enhancement effect. In red drop, we can write that when a when monochromatic beam of 700 nanometer or greater than 680 nanometer only is given the reported minimum photosynthetic yield. Means when we give only one light continuously, you will not see a too much photosynthetic yield. You will see the minimum photosynthetic yield. Means in the presence of only PS1, 7 means only PS1 you will see. Means red drop is also observed in absence of PS2, we can say. Yes or no? We can say that it is occurring in the absence of PS2, and that is a red drop. Means its yield is not that much as much it can be possible. But in immersion enhancement effect, if we talk, you will see that they explain that when both 700 nanometer, that is PS1 and light, and 680 nanometer, that is PS2, both are applied together. They reported enhancement in photosynthetic yield. They reported enhancement in photosynthetic yield. So when we supplemented PS1 with the PS2 or 700 nanometer with the 680 nanometer, the photosynthetic yield increased. This shown that in yield, the enhancement was observed. Because of this, it is called as immersion enhancement effect. So this is what about we can say the red drop immersion enhancement effect and the non-cyclic photophosphorylation. Okay. Now, so this is what a non-cyclic. Now, this which we have discussed a non-cyclic. Okay, P680 pheophytin electron flow, then light photolyzer water electron provided. Okay, then PQ cytochrome B6 and F form cytochrome complex. Plastocyanide is there. Ultimately, electron which is moving from PS1 that is involved in NADPH. This is electron transport chain. This is also electron transport chain where electron carriers are involved in the transfer of energy. Now here, the electron which is present in the magnesium of this P680 and P700, when this get energy, it is reached to its higher excited state and released from the outer orbital. So energy here in the electron is lower, but here it is higher in the upper in this pheophytin and the electron which are moving here. Okay, so this is the what ultimately a diagrammatic representation of this non-cyclic photophosphorylation. Now in this, the NCRT, this diagram is given where you can see LSC of PS2 is there, the electron acceptor is there, that is pheophytin ATP synthesis electron transport system, light heuristic complex of PS1, then electron acceptor of PS1, and here NADPH synthesis. This is the photosystem 2, photosystem 1, light is provided to both, and this photolyzer water associated with the PS2. Okay, and that particularly carry out oxygen evolution. So this is the NCRT diagram that we have understood. Now, what is thing means that we have discussed in the previous slides. Now, here, next thing in jet scheme or a non-cyclic photophosphorylation, we need to understand that is a redox potential scale, or we can say redox potential. So when we talk about a redox potential, the first question is what is it? Then it is defined as measure of the tendency. It is a measure of the tendency of a chemical molecule to acquire electrons and thereby get reduced. So it is a tendency measurement of a chemical molecule to acquire electron. What is the capacity of to acquire electron and get reduced? That is a redox potential. Now it is also called as oxidation reduction potential. Oxidation reduction potential. Okay. Now it is measured. It is measured in watts, that is V, or millivolts, that is MV. Okay. So this way, this is a redox potential. So it is a capacity or tendency to acquire electron and get reduced. What is the capacity of to get reduced? That is the redox potential. So if we talk about this redox potential scale, and if we discuss this Z scheme with the redox potential, if we see, this is a PS2, this is PS1, this is the excited state of chlorophyll or their electron acceptor. This is excited state of PS1 or P700, and this is the particularly their acceptors and NADPH synthesis. Okay, now you can see this is the redox potential scale in electron volts. It is given here. It is 0.0, minus 0.4, minus 0.8, minus 1.2. Here 0.4, 0.8, and 1.2 is given. Means if we talk here, this chlorophyll molecule in unexcited state, it has a higher redox potential value than the excited state. But the energy show arranged according to energy, it is in reverse state. So according to redox potential said, the capacity to acquire electron is more in unexcited state of chlorophyll A compared to the what we can say excited state. And same way, the redox potential value is getting decreased as we go along the electron transport and increasing as the electron transport. And as it is increasing, because of this, the electron are getting acquired. So means we can say here this has a high redox potential, this is a low redox potential, and as the electron transport came up to the PS1, it is increasing. Then here it has a low redox potential, it has a high redox potential. Then at to NADP there is a high redox potential. So this particular exception occurs. So here redox potential value is high, here low. Then also we call it as a uphill transport of electron according to redox potential scale. And here we use a term downhill. As we grow here, we use a term downhill transport of electron. Here we use uphill transport of electron. Here we use downhill transport of electron. Okay. Now uphill, downhill. Here redox potential is high and here less, so it should be downhill, but we are using the uphill word. Why? Because these terms are used according to the energy scale. Here they have a less energy, here they have higher energy, so it's uphill transport. Here they have excited state of chlorophyll P680 has a higher energy and P700 has a less energy, so it is a downhill. Then this excited state has a low energy, so it is a particularly higher energy, and here low energy in the unexcited state, so it is uphill, and to the NADP also it is downhill. So this way, according to energy, these terms are used, which are opposite to the value of redox potential scale, but the terms are used according to the particularly energy task, and they are explaining this redox potential. So you should remember that this is uphill, this is uphill, this is downhill, this is uphill, and this is downhill transport occurring this. So in whole scheme of electron transport of electron, starting from PS2, the electron are transferred uphill to the acceptor, that is P of IT, then down to the electron transport chain to the PS1, that is downhill transport, then from PS1, etc. So another electron finally down, uphill, and then finally downhill to NADP and reduction to the NADPH2 takes place. So this whole scheme gives a Z scheme due to its characteristic shape. Okay, and thus the shape formed when all the carriers are arranged in a sequence on the redox potential scale. Okay, so this is what about redox potential scale for the non-cyclic photophosphorylation that we have discussed. Now, after this, we are going to discuss the cyclic photophosphorylation. And dear students, in cyclic photophosphorylation, cyclic means from where the electron are released, they will come back to it. Okay. So let's discuss cyclic photophosphorylation, where electron flow will not in a non-cyclic way, the electron will is released from where it will come back again to that position. So we calling it as a cyclic photophosphorylation. And here also ATP synthesis is going to take place. So in cyclic photophosphorylation, if we talk, only PS1 is involved. And if only PS1 is involved, these are its accessory pigments. And this is chlorophyll A, that is P700. Now when light reaches here, the ultimately light is reaching here, and from here the two electrons are lost from the magnesium of a chlorophyll A or reaction center of this PS1. This electron are received by iron sulfur protein. Now iron sulfur protein gives its two electron to ferredoxin, and ferredoxin will gives its two electron to plastoquinone in place of NADP. This plastoquinone will gives its two electron to the cytochrome B6, then to cytochrome F, then to plastocyanine electron carrier, and ultimately to the magnesium of the chlorophyll A of PS1. So this way, the cyclic flow of electron is occurring, where the electron which is lost from the magnesium of chlorophyll A of PS1, that is coming back to this again magnesium, and during which the energy is released, and that is used in the phosphorylation of ADP and synthesis of ATP. Because of this, we are calling it a cyclic photophosphorylation. We are calling it a cyclic photophosphorylation. Now the question is, what we understood by studying the cyclic photophosphorylation? So in that, we understood that in cyclic photophosphorylation, if we talk here, only PS1 is involved. Then if here only PS1 is involved, only ATP synthesis occur. Then only PS1 is involved, only ATP synthesis occur. Okay. Then NADPH2 synthesis are not occurring. Then no photolysis of water. No oxygen evolution. So only PS1 is involved, only ATP synthesis, no photolysis of water, no oxygen evolution. So this way, how it differ from the non-cyclic. In non-cyclic, we have to study both PS1, PS2 are required, ATP and ADPH2 both synthesis occur, photolysis of water also occur, and oxygen evolution also occur. That is not occurring in case of the cyclic photophosphorylation. Now we have to last understand what is the location. So in this, only PS1 is liquidated, and PS1 we know it is present on non-oppressed region of both stroma and grana thylakoid. So its location is membrane of both grana and stroma thylakoid. Membrane of both grana and stroma thylakoid. Okay, in case of this. Now, this is cyclic photophosphorylation that we have understood here. And what we are getting from the cyclic, that also we understood. Now, this cyclic photophosphorylation, according to NCRT, the diagram is shown like this: that P700 will receive the light, then electron excitation will occur, electron will be accepted by electron acceptor through electron transport chain. The particularly electron will come back to the PS700 and ATP synthesis. So this is what a cyclic photophosphorylation and how in this way it is differing. Okay, this is differing in this way. So that is all about the cyclic and non-cyclic photophosphorylation and how this cyclic and non-cyclic photophosphorylation occur that we have discussed. Now, in this, the question is in photophosphorylation that we have written in both non-cyclic and cyclic photophosphorylation that the ATP synthesis occur near about plastoquinone. Why ATP synthesis occur near about plastoquinone? How this ATP synthesis during this non-cyclic and cyclic electron flow that we have to understand. And for this, the another theory was given by one of the important scientists that is Peter Michel, that is chemi-osmotic hypothesis. So we will discuss this chemi-osmotic hypothesis in the separate lecture, and that will help us to understand how ATP synthesis occur during the both cyclic and non-cyclic photophosphorylation. Okay, I hope you understood in this lecture. Till that, thank you everyone.